Good day everyone. We will now discuss a new topic, which is how do we diagnose disorders that result from a missing or additional chromosome. This is the basis of the chromosomal karyotypes. So these are the overview of the topic. So by definition, the chromosomes is a genetic structure of cells containing the DNA. For identification of chromosomes, we have to note that each chromosome has a characteristic length and binding pattern. So each autosome is numbered from 1 to 22, and the sex chromosome can either be X or the Y. So in here, this is the parts of the chromosome. So we have here the P arm or the short arm. We have here the centromere, which is the constricted portion of the chromosome. This is important for mitosis or for cell division because it contains the microtubules for mitosis, which attaches the kinetochore. And then this is the long arm or the Q arm of the chromosome. How do we label chromosome? Now we know that the chromosome is identified with a number ranging 1 to 22, or for the sex chromosome, X and Y. Each arm is divided into subregions and is identified by a number. And then each subregion is divided into bands identified with a number. Example is this one, 1q2.4. So this means that we identify the first chromosome, the long arm, because it is Q, the second region, 2, of the chromosome, and the fourth band of that subregion. So that's 1q2.4. Another example is this one. This should be 1q1.3. So this is the first chromosome, and we identify the long Q and the first region, subregion, and the banding of that specific region is on the third. So that's 3. So 1q1.3. A karyotype, by definition, is a photographic arrangement of complete set of chromosomes of a cell or organism. So this one. So 1 to 22 is the autosome, and then this one, the X or Y for the sex chromosome. How do we obtain a sample for karyotyping? So we can use a fecal sample for karyotypes. So commonly used is number 1 is amniocentesis. So a sample is taken from a fluid of the amniotic sac. Or we can use a chorionic villus sampling. So the sample is taken from the fetal tissue that forms part of the placenta. So chromosomes are stained for easy visualization, and we can use a light microscope to view chromosomes in metaphase. So metaphase meaning of metaphase or cell division of mitosis. Chromosomes are arranged into homologous pairs based on the size or length, the banding patterns, and the location of the centromere. So the staining. The banding patterns can be visually identified on chromosomes after staining. These are the traditional types: the G banding for Giemsa stain, the Q banding for fluorescent stain, and the R banding or the reverse Giemsa stain. The new type of staining is the fluorescence in situ hybridization or the fish techniques. So the classic method, we just stain homogeneous chromosome by Giemsa. So we can use this for mutagenic study. While the banding method, so band, this one is homogeneous, so lahat masistain. This one is the banding. So we differentiate individual chromosomes and their parts based on the bands. So this is for detection of numerical or structural chromosome abnormalities in clinical cytogenetics. So these are the bands, the G bands. So we use trypsin or saline solution. The trypsin, we know that the trypsin will denature protein. So if we denature protein, we leave behind the resistant chromosome. And then we stain that by Giemsa. So there is dark and pale bands. This is due to the AT and the GC portion of that chromosome. So the dark, if we talk about the Giemsa, the dark So this one, the dark will stain the 80 rich part of the chromosome, while the pale bands will stain the GC portion of that chromosome. So the GC we know is a hydrogen bond type of bond. And then for the reverse banding, this is just the reverse of the G band. We use a saline solution at high temperature to denature the proteins. So in here, the dark is due to the GC rich components of the chromosomes, and the pale is due to the 80 rich compound of the chromosome. And then for the C banding, we use a strong denaturation of euchromatic parts, meaning we denature the euchromatin. When we say euchromatin, this is this is transcriptionally active part of the chromosome. So we can use a hydrochloride or barium hydroxide plus saline solution at high temperature to denature the euchromatins, leaving behind the resistant heterochromatin, which will stain dark. And the strongly denatured euchromatic parts are pale. So this is for the study of the heterochromatic variants. So this one for the banding methods, so the G bands, we use a trypsin solution with buffer so that we can lyse the protein and then staining it with Giemsa solution. So this is the G band. Take note that there are darker stain. This one, yung mga dark. The darker stain is again because of the 80 rich compound. The paler is due to the GC bonding. So we know adenine thymine and the guanine to cytosine na complementary base pairing. For the R banding or the reverse, take note that this is just a reverse. So this one, the dark, is due to the GC portion. So the darker state, the reverse banding can be used to detect mga centromere or telomere part, because the telomere is the inactive portion, the inactive part, transcriptionally inactive because it is at the end. So end na yung telomere. So the reverse banding can be used to stain the telomeres darker. And then for the C banding for the heterochromatic variants. And this is for the fluorescent staining. So how do we make a karyotype? So if we use a blood, we can draw a blood and then add a few drops and then add phytohemagglutinin to stimulate mitosis and then incubate at 37 degrees for 2 to 3 days. And then we add colchicine or colchicine. The colchicine stops the mitosis in metaphase. So this is important. And then transfer the cells to tube and then adding fixative and then centrifuge and staining and then adding it to the microscope slide and staining it with a Giemsa. Then examine it with a microscope. In other perspective, if we use a tissue sample, then we culture it in a growth medium and then we add chemical to stimulate mitosis and then incubate for 2 to 3 days. And then we add chemical to stop mitosis in metaphase. The most common is the colchicine. To stop mitosis at the metaphase. And then transfer the cells to tube and centrifuge to concentrate in layers. And then adding fixative and putting it in a microscope slide and adding a stain, the Giemsa stain, and then identify and photograph the chromosome. The information obtained from a karyotype is we will know the number of the chromosomes, the sex chromosome content, the presence or absence of individual chromosomes, the nature and extent of large structural abnormalities. The karyotypes can show the chromosomal makeup of an individual. So if we know the number of the chromosomes, then we can identify chromosomal variations that can cause genetic disorders. Now we proceed to fluorescence in situ hybridization, or the FISH technique. So we've done with the traditional or the classic method of staining the karyotype the chromosomes. Now we proceed to this new technique, the fluorescence or the FISH. The FISH hybridizes a DNA nucleic acid probe to a target DNA sequence contained within a cell nucleus. So a variety of specimen types can be analyzed using a FISH. So the intact cells are attached to a microscope slide using standard cytogenetic methods. The FISH is used for chromosome microdeletion detection, interphase chromosome enumeration, gene rearrangements, cryptic chromosomal rearrangements, marker chromosome identification, and chromosome breakpoint mapping. So this one, we can use FISH for detection of single to multiple genetic events. So pag one color, we can see single target. Pag two colors, we can see dual targets. And then multiple targets, it can show multicolors. So this allows one to look at multiple genomic changes within a single cell without destruction of the cellular morphology. So what are probes? So in FISH, we use probe because a probe is a nucleic acid that can be labeled with a marker which allows identification and quantitation. This will hybridize. When you say hybridize, it will attach, it will bind to another nucleic acid on the basis of its base complementarity. So we know that the DNA follows a base complementarity. So A, T, and then G, C. So a probe can be a radioactive, attached labeled with radioactive, such as this one, or the non-radioactive isotope, such as the FISH or the fluorescent, meaning the probes are attached or labeled with a specific isotope so that it will light up, it will color, we will identify. Example, the fluorescent, it will light up if the probes will attach that specific nucleic acid, it will light up. So we will see that in a fluorescent microscope. Or we can also use this one, biotinylated or avidin streptavidin, as a label of probe. So for emphasis, a probe is a part of DNA, or it can be an RNA that is complementary to a certain sequence on target DNA. So that is the DNA of the patient. The probe can be a plasmid, a phage DNA, a cosmid. This is a combination of phage and a plasmid DNA. So it can also be a PCR product. When you say PCR, it is a product of amplification of a certain segment of chromosomal DNA. PCR will be discussed later. So this one is the direct fluorescent labeled probe. So if we have here the specimen DNA, this one is the specimen. This is your target DNA. Then the FISH probe DNA, the one that we will use to bind to that specific specimen DNA, will bind covalently to that of the specimen DNA. And then you have here the fluorescent. The fluorescent is attached. So nandyan na siya so that it will light up. It will color. So after attaching, it will color. So this one, this is the target DNA, and then this one is the probe DNA, the target and then the probe DNA. Both of these will have to be denatured. So ang DNA probe is denatured pa din. So the probe DNA is labeled with fluorescent molecules so that it will color, or we can visualize it. So this one, after that, it will hybridize. So after denaturation, it will hybridize. Hybridize, it will attach so that we can visualize that. There are different types of FISH probes. We can use the centromere, the telomere, the whole chromosome paint or the locus or specific lang. Centromere, this one, the constricted portion. The locus, specific lang siya for that certain region. The telomere at the end of the chromosome or the whole na talaga. Whole chromosome paint. So, this one, centromeric lang siya, ayun lang, part. Or the locus, dito lang din siya. Certain part of the chromosome. Whole, ito na, lahat na siya nagla-light up. All of the chromosome. So, for emphasis lang, telomeric probes have specificity for a single human chromosome or they contain a locus estimated to be within 300 kilobyte of the end. So, at the end lang siya of the chromosome. The whole chromosome painting probes hybridizes probe fluorescent with bright intensity along the length. So, along the length of the chromosome. For the CEP or the chromosome enumerator probes or the centromere area, so they are, most are alpha and satellite probes. So, the centromere region are stained brighter. So, yung centromere yung focus mo. Which means they are rich in the 80 bonds because the centromere is rich in 80 bonds. And then the locus-specific identifiers. The locus-specific is used for mga deletion, microdeletion, translocation. So, it depends on what do you want to use. So, we can have the deletion probes, the translocation probes, the gene detection and localization probes, the gene amplification probes. So, in which condition we have to indicate fish analysis? So, the material when the material doesn't contain a metaphase chromosome, such as in unsuccessful cultivation. So, kasi diba yung cultivation can take up to 10 days. So, if the cultivation is not successful, then we can use the fish. Or, it isn't possible to cultivate the tissue from patient. Example, we need to have a rapid identification, such as in rapid prenatal examinations. We need to get the sample and then we examine the sample quickly. So, in fish, we don't need cultivation pa. So, it takes less than 24 hours for the result to be made. And the analysis of complicated chromosomal rearrangements. We can also use fish for identification of marker chromosomes, for analysis of low-frequency mosaicism, for diagnosis of submicroscopic or cryptic lang na chromosomal arrangements such as in microdeletion syndromes, or amplification of oncogenes and microdeletion of tumor suppressor genes in malignancies. There's also the term multicolor fish. The multicolor fish can provide colorized information relative to chromosome rearrangements, especially useful in specimens where chromosome preparations are less than optimal for standard cytogenetic banding analysis. So this one, ayan, different colors na. So multicolor fish. For the fish procedure, first we denature the chromosomes, and then we denature the probe. Denature mo yung both. Meaning denature, you have to break up. So, sisirain mo yung dalawang complementary strand. And then, now we do hybridization and then fluorescent staining because attached naman sa probe yung fluorescent so that we can examine it on a slide. So, this one, this is the probe DNA which will be denatured, and then this one will be attached to the complementary strand of the DNA or of the target DNA. So ayan, from the tissue sample on slide, if we magnify this, we can view the chromosome. If we further magnify this portion, so we see that the probe will attach to a section of the DNA molecule. So this one, at ito yung probe natin. And then the fish probe, because of its inherent or attached na fluorescent, it will light up or signal. It will light up, or we can visualize this in a fluorescent microscope. So for a better, simpler picture representation, so we can see this one, the target DNA, it will be denatured. Same as the probe. The probe is attached with either a radioactive isotope or a non-radioactive isotope. If this is a fluorescence, then we can see this in a fluorescent microscope. So after denaturation, then hybridization happens. So binding of the probe to that of the target DNA. So, in nucleic acid hybridization is the formation of a duplex complementary to complementary sequences, between two complementary sequences. So, it can be DNA to DNA, RNA to DNA, or RNA to RNA. Annealing is another term used to describe the hybridization of two complementary molecules. So, this one is an example of an automated hybridization. So, this is a hybrid. So, the probe and target DNA are denatured altogether na. So, this can give us a faster, easier, and safer hybridization technique. So, for the visualization of probe, again, it is the DNA probe is labeled with colored fluorescent molecule. So, this fluorescent molecule remains attached to the DNA during the hybridization process. So, the molecule emits a particular color when viewed through a fluorescence microscope that is equipped with the appropriate filter sets. So, this is a fluorescent microscope. So, we have here the CCD camera and then the filters and of course the software, this one. So, here we can see the different chromosomes. So, the difference between fish versus the karyotyping, in here we can see colored siya, no? While in karyotyping, ano lang siya, pale or dark bands lang makita. The result, less than 24 hours, we can expect result na for fish. While in karyotyping, it can take up to 7 to 10 days because we need to cultivate pa the cells. Okay, we're through with the fish. We will proceed to the polymerase chain reaction. The PCR means to amplify a particular piece of DNA. Amplify means making a numerous copies of a segment of DNA. So the PCR was invented in 1984 as a way to make numerous copies of DNA fragments in the laboratory. Its applications are vast, and PCR is now an integral part of molecular biology. First, for DNA replication versus the PCR is a laboratory version of the DNA replication because the laboratory version is commonly termed in vitro since it occurs in a test tube. In vivo signifies occurring in a living cell. So when you say in vitro, outside the human body, so or the living cell. So in vitro fertilization is fertilizing the egg and the sperm outside. So that's the difference between in vitro and in vivo. So in vivo, DNA replication in cells, this occurs it can take up to few hours to copy all of its DNA, and the DNA replication is semi-conservative. The process occurs with very few errors, and more than a dozen enzymes and proteins participate in DNA replication. These are the key enzymes involved in DNA replication in vivo: the DNA polymerase, the DNA ligase, the primase, helicase, topoisomerase, and the single-stranded binding protein. Just to have a quick recap, we know that the DNA polymerase, this one, the enzyme, is responsible to catalyze the elongation of the DNA by adding nucleoside triphosphates to the 3' end of the growing strand. So the DNA polymerase can only add nucleotides to 3' end of the growing strand. So it elongates. The DNA polymerase catalyzes the elongation of the DNA via the complementary base pairing in DNA. So we know that the nitrogenous base adenine will only pair to thymine, and that the nitrogenous base guanine will only pair with cytosine. The DNA ligase is responsible for the joining of the Okazaki fragment in the lagging strand. The primase initiates the synthesis of the DNA. So the primase is an enzyme that can start an RNA chain from scratch, and it creates a primer. The helicase unwinds and twists the two parallel DNA. The topoisomerase relieves the stress of this twisting, and the single binding protein binds to and stabilizes the unpaired DNA strands. What happens in PCR or in the in vitro version of the DNA replication? These are the components needed to perform the PCR in the laboratory. You need the DNA or the DNA of interest, the heat-stable DNA polymerase like the Taq polymerase, because the PCR is temperature dependent, so we need heat-stable na DNA polymerase. And then all four nucleotide triphosphates, the buffers, the two short single-stranded DNA molecules that serve as a primers, a tube, thin-walled tubes, and the thermal cycler. This thermal cycler is a device that can change the temperatures dramatically in a very short period of time. This is a PCR thermocycler. So it contains now the DNA polymerase, the buffer, the nucleoside triphosphates, the primers that are placed in a thin-walled tube and are placed in this one, the thermocycle, the PCR thermal cycler. The three main steps for PCR. So, before we proceed, the basis of PCR again is temperature changes and the effect that these temperature changes have on the DNA. So, in a PCR reaction, the following series of steps is repeated 20 to 40 times. Take note that 25 cycles usually takes about 2 hours and amplifies the DNA fragment of interest 100,000-fold. So, the steps, number 1 is to denature the DNA. So, at 95 degrees Celsius, the DNA is denatured. So, meaning the two strands are separated. Step 2, the primers will anneal. So, at 40 degrees Celsius to 65 degrees Celsius, the primers anneal or bind to their complementary sequences on the single strands of DNA. Next step is the DNA polymerase extends the DNA chain. So at 72 degrees Celsius, the DNA polymerase extends the DNA chain by adding nucleotides to the 3' ends of the primers. So given that the PCR involves very high temperature, so it is imperative that a heat-stable DNA polymerase can be used in the reaction. So, most kasi of the DNA polymerase would denature kasi protein din naman ang mga polymerase. So, we need to use a heat-stable polymerase. So, we can use the Taq, this one, the purified from the hot springs bacterium Thermus aquaticus. So, Taq from the Thermus aquaticus. So, the Taq has a maximal enzymatic activity at 75 degrees Celsius to 80 degrees Celsius and substantially reduced activities at lower temperature. So, mas grabe yung activity niya if mas higher yung temperature. So, this one, the denaturation of DNA. So, this occurs at 95 degrees Celsius, mimicking the function of the helicase in the cell. So, diba? Unwinding of the DNA. So, at 95 degrees Celsius, we just denature the DNA. And the next is annealing or binding of the primer. So, the primers bind to the complementary sequence on the target DNA. And that primers are chosen such that one is complementary to the one strand at one end of the target sequence, and that the other is complementary to the other strand at the other end of the target sequence. And then step 3 is the extension or primer extension. So the DNA polymerase catalyzes the extension of the strand in the 5 to 3 direction starting at the primers, attaching of course the appropriate nucleotide. So the adenine to thymine and the cytosine to guanine. And then the next cycle will begin by denaturing the new DNA strand formed in the previous cycle. So, ito na, may nagawa na tayo. This one, this one. So, after that is i-denature na naman. The cycle will begin na naman. So, by power of 2. So, the size of the DNA fragment produced in PCR is dependent on the primers. So, the PCR reaction will amplify the DNA section between the two primers. So, if the DNA sequence is known, primers can be developed to amplify any piece of organism's DNA. So the DNA of interest is amplified by a power of 2 for each PCR cycle. So for example, if you subject the DNA of interest to 5 cycles of PCR, you will end up with 2 raised to the power of 5 copies. If 40 cycles, then you will end up with 2 raised to the power of 40 copies of DNA. So the PCR has become a very powerful tool in molecular biology. We can start with a single sperm cell or strand of a hair to amplify the DNA sufficiently to allow for DNA analysis and a distinctive band on an agarose gel. You can also amplify fragments of interest in an organism's DNA by choosing the right primers. And then one can use the selectivity of the primers to identify the likelihood of an individual carrying a particular allele of a gene. So the PCR primer are short, single-stranded DNA molecules. So they are manufactured commercially and can be ordered to match any DNA sequence. So the primers are sequence specific. They will bind to a particular sequence in a genome. So as you design primers with a long length, the primers become more selective. So it's more specific. So the DNA polymerase requires primers to initiate the replication. So in here, the selectivity of primers is based on the composition of the sequence. So for example, if only 3 letters lang, the ACC, it would be very likely to encounter its complement in a genome. However, as the size of the primer is increased, the likelihood of, for example, a primer sequence of 35 letters repeatedly encountering a perfect complementary section on the DNA become remote. So very selective ang primer and very specific. So the probability in here, there are 4 bases in the DNA molecules, the ACGT. The probability of encountering any of these bases in the code is 0.25. So 1 in 4. So let us look at this probability. For A lang, one molecule, we can have 0.25 or 25% chance. For 2, kung 80, then we can have 0.0625 or 6% chance of having that 80 na makakuha. If, for example, this one, 16 na. So, the 16 bases in this particular sequence, so the chances is 1 in 4.3 billion. So, in this same way, we can see that as the primer increases in size, the chances of a match other than the one intended for is highly unlikely. So, the PCRs can detect certain diseases or can diagnose certain diseases such as Huntington's disease, cystic fibrosis, and HIV. So we know that the Huntington's disease there is a mutation with the HD gene. In non-HD individuals, the HD gene has a pattern called trinucleotide repeats with CAG or the CAG occurring in repetition less than 30 times. However, in HD individuals, the CAG trinucleotide repeat occurs more than 36 times. So, the PCR can be performed on these individuals' DNA to determine whether the individual has Huntington's disease. So, it is amplified, the DNA is amplified and sequenced. So, a sequence is a technique by which the exact nucleotide sequence is determined and the number of the trinucleotide repeats is then counted. If more than 36, if more than 40, then the patient is more likely to have HD or HD gene. For the cystic fibrosis, there is a mutation in the CTFR gene. So, the presence of the CTFR mutations in an individual can be detected by performing the PCR and sequencing on that individual's DNA. For the HIV tests rely on PCR with primers that will only amplify a section of the viral DNA. So, yung viral DNA, yung amplify niya, found in an infected individual's bodily fluids. So, therefore, if there is a product, PCR product, then the person is likely to be HIV positive. If there is no PCR product, the person is likely to be negative for HIV. So PCR can be used for forensic science. So the application of this is broad spectrum of sciences to answer questions of interest to the legal system. This may be in relation to crime or to a civil action lang. So example of application to the forensic science is the paternity suits and to identify badly decomposed bodies, or when only body fragments are found, such as in World Trade Center, Bosnian, Iraq, and Rwandan mass graves. So that's the last slide. Thank you very much for listening and have a great day ahead.